you gave me a title that I felt like I needed to follow up on and provide a joke to get those laughs. So, two cormorants swim into a bar. They look and notice in the corner there's some sketchy looking fish hanging out. The floor is soaking wet, and the drinks, they're really watered down. One of the cormorants looks at the other cormorant and says, "Geez, what a dive." So the official title of my talk is Relating Seabird Foraging Rates to Spatial Patterns of Juvenile Fish Abundance, or as I prefer, Fishy Secrets Revealed by Watching Seabirds Eat. Quick going back, my co-authors are Dan Robinette, Nadav Noor, and Jaime Yankee. I know Nadav is here today. I think Dan is at a conference. So I'm not sure where Jaime is. Hopefully having a good time. Anyway, there are a lot of fishy secrets out there, but if you understand the environmental influences on fish populations, you can learn some of them, like the reasons behind local population structure and variability, how variability affects marine predators, and if you're interested in marine protected areas, how to create realistic expectations for how fish populations will react when protections are added or taken away. One way that you can learn these fishy secrets is to physically sample the population. However, juvenile fish are small and sampling them is very difficult. Most sampling difficulties are logistical. I mean, the ocean's a big place. Fisheries sampling vessels are usually large and can't come close to shore in shallow waters. Diver surveys are great fun, but they're incredibly labor intensive and they only cover small areas. And juvenile fish are simply harder to find because of their size. So if there was just some sort of specialized tool that we could use to sample. Oh, I think there is one. Let's let the seabirds do the work for us. All we have to do is sit there and watch them. That's pretty easy. So what I'm going to talk about today is 4 related studies that are looking at seabird foraging patterns as they relate to juvenile fish abundance along the coast. The general question in common to all 4 of these papers is, what does seabird foraging tell us about juvenile fish abundance? We looked into the where, when, why, and who. And we found some variable and occasionally unexpected results. But when you're studying fish distribution without actually looking at the fish, you never know what you're going to find. So the theory is simple. Where there are seabirds eating, there are fish. But the reality is actually much more complex. Some of the actors in this work are the fish themselves, but they're really the wizard behind the curtain. We can't see what they're doing, but they affect and are affected by everything around them. The seabirds are the charismatic stars of the show. They're easy to observe from the coast, which makes them a prime candidate for these efforts. The static and environmental features, such as coastal geography, how the wind interacts with the coast, and upwelling strength and variability also influence fish and seabird populations, so those need to be taken into account. Finally, marine protected areas are like the theater in which all of these actors play their parts. A well-designed and managed show is a win for everybody. So I wanted to touch a little bit more on upwelling for those of you who are less familiar with the marine environment, and also some terms of coastal geography, since I'm going to refer to these things quite a bit in my talk. Prevailing coastal winds will drive upwelling. On the California coast, these winds are predominantly from the northwest, and through a process called Ekman transport, they move the ocean surface waters away from the shore, and in doing so, pull cold, deep, nutrient-rich water to the surface near shore. These nutrients are the base of the food web, and they support phytoplankton, which in turn supports zooplankton and larval fish, as well as all the other marine animals, not just mammals. The shape of the coast also affects behavior of currents, and areas upwind or windward of a coastal promontory might have different upwelling characteristics than areas at the exposed tip or downwind, also known as leeward from the promontory. Windward of promontories, upwelling tends to result in offshore transport. So if the upwelling is strong or persistent enough, basically the fish larvae can get washed away from shore. We would expect that in times of persistent upwelling, that there would be smaller fish populations in those locations during the following year, as the fact that those larval fish got washed away. If upwelling is variable or pulsed, those larvae and juveniles have a chance to remain in the environment, as well as occasionally receive influxes of nutrients. Leeward of promontories, the shape of the coast allows for the formation of something called an upwelling shadow, which is an area where we would expect larvae to be retained. The strength and persistence of upwelling has an influence in this area, but in general, these areas are expected to support larval fish and potentially have larger populations. So we're going to try to figure out the influence of upwelling strength and persistence, as well as that of coastal geography on fish populations. So in the first of these 4 papers, we looked at the foraging rates and diet of one seabird species in 2 locations near Point Arguello in Santa Barbara County. We recorded observations of what these birds were eating and where they were eating it over 6 field seasons. The seabird that we were studying was the pigeon guillemot, and it ate different species depending on where it was foraging. In this area, sand dabs, which are a flatfish, were captured by pigeon guillemots more often than other species. Birds that traveled windward of Point Arguello captured a variety of species, with some sand dabs included. But birds that foraged leeward of the promontory captured mostly age 1 sand dabs. During the 6 field seasons, there were changes in the upwelling regime in the area. When upwelling was more variable, essentially turning on and off, guillemots captured more sand dabs in the windward habitats. This indicates that intermittent relaxation in upwelling may allow sand dabs, which are a bottom-dwelling fish, to settle in and mature in these windward areas. However, we also found that guillemots captured sand dabs leeward of the promontory when upwelling was persistent or stayed on. So basically it didn't fluctuate, it was consistent. The shape of the coast in this area most likely has created a retainment area that buffers the effect of continuous upwelling. So this first paper revealed the fishy secret that upwelling variability is important, and that the effect of that variability on juvenile fish differs based on coastal geography. For our second paper, we decided to expand on this study a little bit. And looks like, did that advance? Oh, I'm sorry. I forgot about the second point. The second point that we found on this paper was that foraging and recruitment rates were higher and less variable leeward of the coastal promontories. So we saw definitely that the guillemots were taking more flatfish down there, and they were able to do that consistently over the 6-year study. So our next paper is based on work done in approximately that same area, but with 4 species of seabirds and the additional factor of the presence of a state marine reserve. At the time this study was completed, the State Marine Reserve encompassed the Point Arguello promontory. Since then, it has been expanded to the north. One purpose of the reserve was to protect fish and invertebrate species that are harvested by people. This area is known for notoriously rough seas and shipwrecks, so assessing how fish populations are changing within this protected area is very difficult. Here again, we use seabird foraging rates to try to assess fish abundance. In addition, this reserve may also provide some protections to seabirds. There are 2 major breeding colonies for cormorants within the reserve. And these protections may help reduce bycatch, disturbance, and as well as reduce competition for prey. Of the 4 seabirds we looked at, brants and pelagic cormorants and Pacific loons are all piscivores, and surf scoters are molluscivores. That's always a fun one to say. The 4 study sites were distributed windward and leeward of the promontory and inside and outside of the reserve. So we again had 6 years of data for this study. What we found is that the seabirds species that we were studying in this case had consistent foraging distributions over the 6 years. When we observed them foraging, they were foraging in the same places in the same densities. However, foraging rates were higher for the fish eaters leeward of the promontory, which on these graphs is in green, and the mollusk eaters or the surf scoters had higher foraging rates windward of the promontory, which is the orange line in these graphs. So in addition, the piscivores tended to forage at the same or higher rates outside of the reserves. So in these graphs, the lines slope from inside to outside, left to right. And we didn't expect that. We thought the reserve would be protecting fish populations and therefore there might be more foraging inside. However, the location of the reserve relative to the general coastal geography may actually be driving the distribution of fish populations, and the shape of the reserve is not so much causing this effect as the coastal geography. So the second paper provided support for our second fishy secret from the first one, namely that many species of juvenile fish appear to have higher and less variable abundance in the leeward of a promontory. So, and that is demonstrated again by the foraging rates of birds that consume a variety of species, in this case, the brants and pelagic cormorants. So now for paper number 3, we went bigger, gotta go bigger. We wanted to see if foraging rates were a good indicator of fish abundance in a large area. So we compared seabird foraging data, in this case, to kelp forest fish dive survey data collected in similar areas. This is the one paper where we actually have some fish information. In this map, it shows the study area. The pale green areas are the marine protected areas. The colored shapes represent where different species of seabirds were monitored, and the black letter number combinations are the locations of the kelp forest surveys. All of these data were collected as part of the South Coast Baseline Marine Life Protection Act assessment, which took place over 2 years. Overall, we found that regional foraging rates for all species of seabirds were positively correlated with the combined density of common fish families observed, such that more seabird foraging occurred where more fish were observed. This is expected, but it's a very general observation. It's like it lumps all the seabirds together with all the fish species. So the extent of this area definitely leads to local differences, and the positive relationship for individual species of birds and fish only held between brants and pelagic cormorants and rockfish. The fact that the relationship between foraging in general and fish density in general is less similar at the site-specific scale, maybe because the fish data were focused so much on kelp forest habitats, while seabirds utilize multiple habitat types. They forage in open water, they forage on rocky bottom, they forage on sandy bottom, pretty much, and they forage in kelp forests. So in addition, the distribution of various bird and fish species does vary over such a large area. For example, terns are much more common in the southern part of the area, whereas cormorants are more common in the northern part. So in this case, using seabirds foraging rates may help provide a more holistic approach to determining juvenile fish abundance over a very large area. Diver surveys are, by nature, limited in scope. They can only cover so much ground or water, as the case may be. In addition, since seabirds use multiple habitats, they can provide information on the condition of the wider system. This in turn may help managers understand what's going on at the community level. So this one provided a new fishy secret. Number 3, in general, foraging rates are positively correlated with fish abundance. So that does seem to be something of a gimme, but it's definitely confirmed now through observation. But what about areas that are outside of the South Coast? There's still a lot of California north of Point Conception. Let's go huge. This 4th paper was our most ambitious paper yet. We scaled back on the number of seabird species, but we decided to look at the entire California coast to try to tease out which factors best explain variability in juvenile fish abundance. We looked at foraging data for both brants and pelagic cormorants at 46 sites coastwide or statewide. The reason we chose brants and pelagic was they were the species that were common for the entire coast. Seabirds at these sites were originally studied as part of the Marine Life Protection Act baseline assessment and monitoring for the Seabird Protection Network. For this study, we wanted to look more at the influence of coastal geography on fish abundance, since we have a lot of variation in promontory versus straight coast and windward, exposed, and leeward areas. So we looked at several geographic variables, including the geographic categories of windward, exposed, and leeward, coast direction, and promontory shape. So the image here is describing or is illustrating some of the things we looked at for promontory shape, being the width of the promontory, which is how far it sticks out, the length of the promontory, which is how much of the coastline is taken up by the promontory. And then we also looked at the aspect ratio, so the ratio of length to width. For upwelling, we included analysis of spring and summer strength and variability simply because we were looking at these species during the spring and summer, and all for the year of the seabird data and also for the year prior. And the theory behind that is variability and strength the year prior is going to affect the larval settlement. And then the following year, the age 1 fish that remain are going to be the fish that get taken by the seabirds. So we also controlled for several spatial and temporal variables because there is a lot of relationships between these various things. Each species had different relationships with the different variables, and I will only be discussing the significant ones. Otherwise, we would be looking at graphs until 3 this afternoon. So in comparison, comparing the foraging rates for the 2 species among the different geographic categories, we found that both species foraged mostly at leeward sites, as we've seen before, but especially the brants cormorants. We expect a higher abundance of juvenile fish at leeward sites due to these areas tending to be retention zones. The influence of promontory size, shape, and direction was different for the 2 species. Foraging rates for brants cormorants increase as the base of the promontory becomes wider or covers a longer distance along the coastline. Promontory length mattered more for pelagic cormorants, with higher foraging rates near short and long promontories. It wasn't just one, but both. In addition, pelagic cormorants foraged more where the coastline was south-facing. Interactions between upwelling and geography also differed between the species. When upwelling strength was low, brants cormorants foraged more at exposed and leeward sites. Brants cormorant foraging was also influenced by upwelling persistence in the prior year. So again, this is where the juvenile fish, the larvae that come in the prior year are then developing into juvenile fish the year of the survey. So the moderate prior year persistence of upwelling led to higher foraging rates at exposed and leeward sites. So if it was moderate, we are assuming that there was actually more fish that remained in the area and were able to get large enough for the cormorants to want to take them. On the other hand, pelagic cormorants, of course, have to be different and foraged more at the windward and exposed sites when upwelling persistence was moderately high the prior year. So basically they like a little bit of give and take in upwelling, but definitely don't want it too much or too little. It's kind of like the Goldilocks bird. So this study confirmed two of our previously discovered secrets, that upwelling variability and its relationship to coastal geography is an important determinant of seabird foraging rates and therefore fish abundance. And also that foraging and recruitment rates are higher and less variable leeward of promontories. It also provided new information on the importance of promontory size and shape and the influence of the strength of upwelling on local fish populations, in addition to the variability. So understanding the interactions between coastal geography and upwelling impacts can definitely help establish realistic expectations for the rates of population change after management actions, such as MPA establishment. For example, MPAs that are established in areas where fish eggs and larvae are retained near shore will likely show higher recovery rates than MPAs established along exposed sections of coast. Also, years of persistent, intense upwelling may slow recovery at sites where offshore transport during upwelling tends to be high. So I would like to put in a plug for Pete. Stay tuned. Pete is going to be presenting on October 5th, Fish and Farallon style, where he's going to talk more about seabirds and fish, where he's got actual fish and poop and barf and just about everything else. So keep an eye out for that one. Put that one on your calendar. I would like to thank all the folks who helped us out and supported us in the data collection for this, and all of our staff, interns, and volunteers who also supported us, and some of whom kept me sane. So thank you, and I will take questions.
Thank you so much, Julie. That was amazing. And all the puns just tickled me so much. Love it.
I hope. Okay, there was another movie reference in there. Somebody, or several movie references. Somebody has to put in the chat what it was.
Okay, movie references. Put it in the chat.
Or movie. I'll give you a hint. A movie series reference.
Okay, think on that. Think on that. Oh, one new message.
There it is. Boom. Meredith.
Meredith's
on fire.
All right, so we're going to move into Q&A. That was a bunch of amazing information. So again, if you would like to ask a question, use the hand raise function, please, and I will call on you. And you can find that in the reactions section. And sometimes some of you might be able to do look at that. It does it automatically. Creepy.
Oh, it did?
Yeah.
Is that like a setting? How do you do that?
Mine didn't do it.
If you go to reactions, there's a recognize hand gestures.
Oh, scary.
Click if you're in that version. But anyways. Okay,
there you are,
do you have a
Rose.
question?
Oh, she put her hand down.
I don't actually have a question. Sorry, I'm just
Okay.
Everybody's just going to try the feature.
Okay, Diane, do you have a real question? Okay, Diane, that's the first question asked.
My mute button here. So, Julie, I don't think I've met you yet. For context, I am not a biologist, so this may be a lower end question. I'm trying to learn more about everything we do here. What was the actual data collected? Was this somebody visually just observing birds? Was there, how did you tell what type of fish they were foraging for? Can you explain that process briefly?
Sure. The majority of data collected and used in this is foraging rates. So what we did was sat on the cliff top for 3 hours and counted birds eating every 15 minutes. So you'd scan the area, and any birds that were foraging, you just record species and number. Most of these birds are divers, so that's how we could determine that they were foraging, because they would dive. For birds that aren't divers, we would look for foraging behaviors, picking at the surface, harassing other birds that had fish, etc. So a lot of this is simple clifftop observation, and there are hundreds and hundreds of very cold and sometimes hot hours of data behind this. It took a lot of people to get all this. In the case of the first paper with the pigeon guillemots, in addition to sitting and watching where the seabirds were foraging, where the guillemots were foraging, and where they were going to and from, we also sat and watched what they were bringing in. So they do this on the farallones too, where they'll sit and look at the nest crevices. These birds nest in crevices, usually in cliffs, and you can position yourself and watch them flying in. They stop at the outside of the crevice generally for a nice photo op. Hi, look at my fish. And then go into the crevice. So that allows you to actually look at and identify what kind of fish they're carrying. So for the first talk, we were actually able to identify what they were eating. For the third talk, the comparison was with fish survey data. So we weren't necessarily looking at what the birds were eating directly, but knowing where they were eating it and getting the data from that location where it was correlated. You had another question? Or did I get them?
No, that was it. Thank you very
Okay.
much.
All right,
Great,
Farallon
thank you.
room.
Yeah. Annie, you're up.
Hi, Julie. Great talk.
Hey, thank you.
I wanted to ask about that result with the cormorants foraging more on the leeward side when upwelling was lower. Did I get that right?
I don't know. I'm gonna have to go back in my talk and remember that, because it's confusing in my head too. So basically, Brandt's cormorants more exposed and leeward when strength is low and upwelling is variable. So your question being why?
Yeah, well, now I'm not sure, because I think what you just said maybe makes more sense. But so when you went through your talk initially, I thought it seemed a little bit counterintuitive. So I wanted to see what your explanation for that behavior was, but maybe now it's not.
Right. So I think it's intuitive to me for the Brandts for sure, in that, you know, the leeward is protected. There's more fish there and more fish variety in those areas. I think maybe the confusing one for me is that for pelagic cormorants, they want a lot of variability, and they want it in windward habitats. So, or at least that's
Julie,
where they
can you show
foraged.
the slide?
Yeah, I can bring it
Maybe
up.
that'd
Hold
help.
on. My computer is asking me to reboot, and I'm going to tell it no.
Oh, mine too. We probably all got the same message.
We probably all got it at the same time. Hold on just a sec. All right. So you should be able to see my screen.
Yes.
Okay. Let me see if I can go back. All right. So here's I think this is the first half of the question for the Brandts. Is this
Right. Yeah. Okay.
Yeah.
I think maybe so they're foraging more at both exposed and leeward when upwelling strength was lower.
Yes.
Okay, I guess that does make sense. I think I missed the exposed part.
Oh, okay. So, but this one in Nadav, maybe you can jump in, other than it's a statistics thing. As like why pelagics were more focused in the windward areas when there was a lot, when upwelling was stable, which would be mostly when the offshore transport is higher. Pelagics do tend to have a slightly narrower diet than Brandts do. They focus on fewer species. So it's possible that the species that they are interested in are intermittently present and they're just hitting them when they show up. So, Annie, if you have any other thoughts on that, I'm happy to hear them.
No,
Yeah, I mean, our results were showing that the difference, as I said, between the two species, and I think it's just interesting in general. I'm not good at parsing out all the individual ones, but yeah, showing the differences, which were, obvious. I think, Julie, that's what you're implying. It isn't like, oh yeah, we have these, great ideas we're going to test out. So it's interesting. I find it interesting, and I don't think Pete is going to talk about it next week, or 2 weeks, whenever it is. But just pelagic and Brandt's cormorant show such different patterns of reproductive success on the farallones, where we have studied it for a long time. So
And
they're
they
different.
do here at Vandenberg too.
Okay. So I think that's really interesting. So we're seeing on the reproductive side, but then also seeing in these foraging differences. Yeah, and I think we need to go a little further. For me, with that study that Juliet summarized, is be great to add some more years, which I guess we have tucked away of data. I think that was only the 2 years, right? Was that the multiple? It depended where. Yeah, sorry.
It depended on where. Yeah.
Yeah,
For
yeah.
Vandenberg,
For the larger
for Vandenberg, it
coast.
was 6, but for everywhere else, it was 2.
Oh yeah. Okay. Well, anyway, there's a lot of things we could still learn.
Yes.
We have a question in the chat
I
from,
see
I think
that.
this is the Palomar Field Station, or Mark, Point Blue. Mark is Point Blue today. What happens to the juvenile fish when they get transported further offshore?
That's a really good question, Mark. We need some pelagic seabirds to give us some information out there. Maybe we can catch some birds during our access surveys while we're far at sea and lavage them and see what they were eating. To be honest, I don't know. I'm sure that a lot, juvenile fish are highly dependent on primary production and to survive. And if they get washed off into an area where there's not a lot of phytoplankton and zooplankton, they probably become food and nutrients that get washed back in eventually for the other juvenile fish. That's a theory. I am not a fisheries person.
So I don't see any other hand raises right now or chat questions. So I'll fill in with a question while people are thinking of their next ones. We got about 10 minutes left for Q&A, so keep them coming. One thing I was curious about, Julie, is like the point at which this data starts to inform the boundaries of the MPA. Like it sounds like the coastal geography is like a kind of an important sort of discovery that's coming up in the studies that you're doing in combination with all the upwelling stuff. So it sounds like the MPAs currently aren't really matching well with those things that you're coming up, or they could match better, and that it might potentially be dynamic. So it kind of makes me think about like when Cotton was presenting about whales and shipping lanes, like it seems like the ideal would be to have dynamic management, but then like the shipping people were like, just give us something to like follow and we'll do it, but we don't want to keep changing it up. So I'm just curious about like how that sort of dynamic works with these data informing MPA boundaries. Like, is there that sort of conflict or tension?
I'd say yes. It is not dissimilar to that, where it's like the hope for this is to have adaptive management of MPAs using this information, which was not necessarily considered when the MPAs were originally designed and placed. I can speak with some authority to Vandenberg's. So Vandenberg State Marine Reserve was relatively small, was established before the MLPA process, but then was expanded to the north with the MLPA process. The boundaries were determined based on the areas that get closed during rocket launches, and therefore essentially with the assumption that Vandenberg would have jurisdiction over these areas. And so therefore the protection would hold because Vandenberg would say, you can't go here. There's nothing biological in that decision. It's purely political. And some MPAs were certainly designed with that in mind, with biological characteristics in mind, some with both. And a few were just like, no, we're going to protect this area because we want to. And it's like, well, is it a good area to protect? We don't know. That's the whole purpose of this MLPA assessment, as it's like been 10 years now, or 5 years, to keep this process, no, 10, because it started in 2012, to keep this process going, to try to learn, are these MPAs doing what we want them to do? And if you look at an MPA that's on an extremely exposed piece of coast and it's not supporting fish populations haven't changed there, well, then why? Is that the MPA's fault? Or is it the fact that the MPA is placed poorly? Or is there something else going on, like all the adult fish around the MPA are being taken because you can't fish in the MPA anymore? So there's so many factors going on. But yes, I think what we're hoping is that these characteristics will be taken into account in the assessment of MPAs going forward, and determining, okay, even if this MPA is not, the fish are not recovering as fast in this MPA as in other MPAs, why is that? Is it geography? Is it climate change? Is it something else that's going on? Does that answer your question?
Yeah, I think it does to a large degree. And I guess one follow-up is like who are you trying to keep out of the MP, to the protected areas? Like, is it fisher people? Is it like kayakers? Like everybody in
different protected areas have different levels of protection. So the most extreme level of protection is basically like, you can't go in there. There's very few of those, but they do exist. A few of them are near seabird colonies, and the whole purpose is to protect that area around the seabird colony, so that to reduce disturbance to the colony, as well as to protect the foraging areas for those birds. Some areas you can kayak in, no problem. You can use it all you want. You just can't extract. So you can't fish, you can't, dive and collect things, et cetera, et cetera. Other areas you can't collect certain species. So they're made to protect rockfish, say. But you can fish for other species in that area. So
okay.
Again, MPAs are complicated.
Yeah.
The coast is complicated. It's all complicated.
So yeah, with a lot of this data that we just heard about, these data, is that what I'm supposed to say?
Dem data.
Dem data. We're trying to figure out how to protect areas so that like the ecosystem can be resilient and continue to provide food, like for certain species of fish and birds, which reflect other animals out there too.
Yes, that is the general basis for marine protected areas, is to protect some aspect of the ecosystem for the future, and also to provide refuge.
Yeah.
But there's different levels of protection. So each various MPAs are going to have different levels of influence.
Right. Corey put something in the chat. I think you may have answered it. Corey, do you have any follow-up questions to that?
No, that's okay. She kind of answered it too. It just sounds like that's all still in process. We're trying to identify those ones that aren't doing as well. Sounds like this is one step in that direction.
Right.
Yeah. Thanks.
I see Mike.
Thanks, Julie.
You're welcome.
My question is, what are your plans to go even bigger with this work? What are the future objectives?
We're going to do seabirds in space. See, we're going to launch some fish on the next rocket, and then tagging would be awesome. Yeah, to be honest, I'm not sure what the next steps are going to be. But I'm more than happy to cooperate with places that can tag, since Vandenberg cannot tag, unless you want to do a full-on Mission Impossible off a cliff. But hey, there's
That could
another
be
movie.
fun.
Yeah, it could be, as long as you don't mind the cliff coming down on you and cutting your rope in the process. But yeah, no forward thoughts yet, but I'm sure we'll develop something soon. And I can tell you about
I thought-
20 seabirds in
Sorry,
space.
Julie, I thought we were going to do, or Dana talked about doing the additional years and sites. So that could result, hopefully, more insights. I mean, I think it's, it's a good start so far. But I mean, the big California coast,
Yeah,
it's a lot.
yeah, it is
I mean,
a lot.
Mike said bigger, but that's the way, I mean, we have some analyses that have already done the entire coast.
Right.
And obviously the region by region, I mean, Julie alluded to that. That was part of the analysis, was to try to control for regional effects. So that would be a start. I was actually interested in timing of that, and I wonder if we can get maybe we can, because in general, like you mentioned, Julie, we looked at upwelling and just sort of what's the spring summer upwelling. But within spring summer, there's variability. And I think there's an opportunity to look at that, because I think that's going to be important for the seabirds, foraging on the fish. Is this year, when was that upwelling pulse and so on?
Sure. And to be honest, the last time, even though some of these papers were just published, the latest version of any of the seabird foraging data was in 2016. So we're 8 years out now. And given that the MPAs are kind of going through this assessment process, if there's any opportunity to repeat these surveys, we could do comparisons between what happened in 2012 to 16 and what happens in 2023 to X. It's like the Farallon room has another question. Annie.
Well, I'm not sure if this really is a question. I guess it's a question. I'm wondering, like, where anchovy fit into the, so are the cormorant, the brants in the bite down there eating anchovy also? Are those close to shore ones not eating anchovy? What's going on there?
No, they'll take anchovy if it's available, for sure. I mean, I've certainly seen regurgitated ones sitting on the nest ledges. How specific species aside from sand dabs fit into this, I'm not very knowledgeable about. We do have a cormorant pellet assessment that Meredith definitely knows more about, and maybe we could do a future talk on that or kind of look into that more. I do know that Pete's going to cover stuff with Farallons for that, but down here, I'm unfortunately less knowledgeable and probably unable to answer your question to your satisfaction.
Okay, no, that's good. Thank you.
In other words, I don't know.
Fair enough.
Sweet. Awesome. Thank you so much, Julie. That was so informative and funny. I started recording after you told the joke, so I might have to have you tell the joke again so I can record it and splice it into the beginning of the recording.
Okay. we'll let everybody disconnect and then we can do that afterwards.
Okay, great. Thanks everybody for joining today. I think we had 22 as our top number participating. That's another great gathering. So thanks
Yes.
everybody for taking
Thank you,
the
everyone.
time
I
to
appreciate
learn.
it.
Join us next Thursday with Nadav talking about untangling whales with science. We'll get more detail on that. It's just a funny title again.
Untangling with science.
And then the next Thursday, we're going to get into the Southern Ocean, some penguin science with Dennis and Susie, and then wrapping up oceans with Pete in October, like Julie mentioned. So we will see you next Thursday. Have a good rest of your day and see some of you at Birdathon kickoff this evening in Tiburon.
Bye. Thank you.
Thanks, Julia. It was great.
Bye.